Okay, so let's continue. So what is going to happen in the Krebs cycle, and what is the point of the Krebs cycle? So what needs to happen now is we need to kind of link the pyruvic acid into the acetyl coenzyme A. It's a kind of very long molecule, so we need to add that pyruvate into the acetyl coenzyme A. Krebs cycle is happening in the matrix of the mitochondria, and the point of Krebs cycle is this is where all this reducing power is being made. Now, for those of you who still don't know what I'm talking about, this is what I'm talking about. So this is the low state of these electron carriers. So once this molecule accepts electrons, and especially electrons as the hydrogen protons, they go from their low onto the high energy state. And now in this form, they can go and carry this energy into other processes which are needed. So as Krebs cycle is happening, we are kind of piling up on this reducing power. So this is a picture of the Krebs cycle. Here you can see the pyruvate, which is being merged into acetyl coenzyme A. So it's a cycle. It really doesn't have the first and the end molecule. And like I said, for a plant phase, we are not going to talk about it, all the details. You see all the amino acids and all the enzymes in here, which convert one amino acid into another. The key points are right here. This is where this is where carbon dioxide is being kind of expelled. And then you can see all these places where we are adding that reducing power here, 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 and right here at the beginning. So what is happening is that in the matrix of the mitochondria now, there will be a lot of that reducing power. Now, I'm just going to go to another picture. I don't really like this one. But so this is the membrane of Christie, right? And then that's the outer membrane. So here would be cytoplasm, the outer membrane, and then the inner membrane of mitochondria, and then the space in between those two membranes is called the intermembrane space. So right here is where Krebs cycle is happening, and of course, there will be a lot of reducing power, which will now go on to the photosynthetic electron transport chain. Photosynthetic electron transport chain is happening on the inner membrane of the mitochondria. So it is happening on the membrane called Christie. So we talk about that. So that's the membrane where you see the electron transport chain. So let me just use this picture. I kind of like it. It's a little bit better. So the starting of the electron transport chain will be when the NADH will give the two electrons and then go from its high to its low energy state. I mean, this image shows one electron, but there are always two electrons which are being shuffled. And again, it's a very similar thing as in photosynthetic electron transport chain, where you see two electrons being shuffled from one transmembrane protein onto the other. So we have also mobile transport carriers. We have a cytochrome complex, similar, not the same as in photosynthetic electron transport chain. Now, in the end of the electron transport chain, something needs to catch that electron, and that something is 1.5 oxygen. So this is why we need to breathe. This is why we need to inhale oxygen. I mean, both we, us, plants, and animals, because we need something to catch those two electrons. So, and that's going to be oxygen. So you're going to have water that is being produced at the end of the electron transport chain. Now, simultaneously, so when, let's take an example of this transmembrane protein. So when it gains these two electrons, when you have two electrons, you can have extra energy to do work, and that work will be to pump hydrogen protons into the intermembrane space. So now there will be a lot of them here and less of them here. So pretty much the transmembrane proteins are doing work because they are pumping hydrogen protons against their concentration gradient. Remember, if there is a lot of hydrogen protons here and less of them in the matrix, naturally they want to go back. They want to pass and go through here to here because they just want to flow spontaneously from the point of their high concentration from the point of their low concentration. But they cannot do that because the plasma membrane on mitochondria is so densely packed that they cannot flow through the membrane freely from the point of their high concentration to the point of their low concentration. So it takes a lot of effort to kind of pump the hydrogen protons where they do not want to go. Now, in the end, right here, you are going to have ATP synthase, which is a molecule that is going to be used to create ATP synthase. So let's talk about that a little bit. The way the ATP synthase works is that it's called ATP synthase. So it's an enzyme that is synthesizing ATP using the energy from a proton motive force. So what is that proton motive force? Let me just go to this image. So there will be a lot of hydrogens right here, and they're going to kind of flow through the ATP synthase, almost like a windmill. And as they go through, the ATP synthase will turn. Okay, it will turn like this, counterclockwise. And the energy that this enzyme is utilizing, right? We said it's not ATP. It's called the proton motive force. So there will be a force of protons which are kind of going down through the ATP synthase and then making it move. That's why the name proton motive force. So as those hydrogens which are being pumped into the intermembrane space, as they flow through, this molecule is going to move counterclockwise. It works very similar to like this water mill, which you see right here. So as the water goes in this direction, then this mill is turning and doing work. Okay, this is very important. And this work that is being done, it is to add this phosphate group onto the ADP. So in this subunit right here, which is in the matrix, it has to force ADP and P together. And I usually ask the students why, like, for example, if you have a jar of an ADP and a jar of phosphate, like, why do you need to merge those two together? And if you look really close, you can see all this negativity. So phosphate and ADP, they kind of repel each other. So you have to use force, okay, to create those high energy bonds. You need to like force these two molecules to join together to create ATP. That's why ATP synthase needs to actually, make a motion, and it has to move to kind of squeeze together ADP and P so that we can have ATP. So what you see in the process of respiration, we have glycolysis happening in the cytoplasm, pyruvate that goes into the Krebs cycle. Then you're going to have electron transport chain, and then you will see, carbon dioxide, water, and of course a lot of ATP, which is kind of being leaching out and transported out of the mitochondria. Krebs cycle, many of those amino acids are used to make some other amino acids, which are then, of course, going to be used to make protein. Okay, so Here you can see that the respiration is coupled with the cell's need. So of course, the more biochemical processes are happening in a cell, if the cell is very active, it's going to require a lot of ATP, and it's going to give away a lot of products, a lot of byproducts of using that ATP. So then the respiration rate will increase. So cells which are metabolically active need to have a lot of ATP. That's why when you study, it's good to open windows so you have that air coming in. Also, when you study, you will kind of crave chocolate, right? That's where, because brain cells are one of the most cells that require most energy. They're very active. They're in the metabolic sense, they're very active, so they require a lot of ATP. So just open those windows, have some fruits, vegetables, and chocolate. So that's going to help you study. And let's just finish up talking about test 3, which is going to be the written assignment on the rest of the topics. I will give more details on Moodle with the deadline and page limits and how it's supposed to look like. Each student will have a different topic, and those topics will be sent on to your UB individual emails. So that is going to be your test 3. That's all the information I have so far. So there will be no more lecturing from me. So all you have to do now is finish test 2, finish the written assignment, and then when I have more information, I will update you on the final examination. And another update is the whole semester has been pushed for a week. So now the end of the semester will officially be the 28th of April. Yes, so not the 21st, but the 28th of April. Okay.